lot of gene regulation in eukaryotes occurs at the transcriptional level. So in the previous section, we saw that we had epigenetic regulation mainly at the chromatin level. Here at the transcriptional level, we're talking about what's controlling RNA polymerase binding to the promoter. So eukaryotes, just like prokaryotes, have to have RNA polymerase bind to the promoter. This is going to involve some proteins called transcription factors. They're kind of like the sigma factor, but there's a lot more of them. So we'll talk about the role of transcription factors, and then we'll talk about these DNA elements called enhancers and repressors. So there's two types of transcription factors. There's general transcription factors. These are important in getting RNA polymerase to what we call the core promoter element, the bit that's right in front of the gene, and they help RNA polymerase bind there. All genes are going to use these general transcription factors. There's also specific transcription factors. They're going to bind to areas outside the core promoter on the DNA, and they're going to interact with the general transcription factors to either enhance or repress the level of transcription. So these transcription factors are more gene specific. You might have one transcription factor that helps control the expression of a couple hundred genes. Now, it sounds like a lot, but when you have 25,000 genes, that's pretty specific there. So how do they do this? How do they influence this rate of transcription? Well, they bind to elements of DNA called enhancers. Enhancers are DNA sequences outside of the core promoter that are bound by specific transcription factors. So here we have two genes, gene A and gene B. And here's the core promoter here, where RNA polymerase binds with all of the general transcription factors. Then you can see further downstream in the DNA, we have these what we call enhancer regions. They're little bits of DNA that are bound by transcription factors, the specific transcription factors. So these enhancers are far away. So how do they interact with RNA polymerase? Well, there are DNA bending proteins that allow the DNA to fold over. And you can see that these activators, as they are called, these transcription factors, are interacting with the RNA polymerase. So they can increase the rate of transcription or they can decrease the rate of transcription. That gives us a lot of control. Instead of just being off or on, we can have it a little bit on or a little bit more on or more, up and up, right? So we can further control this. Now, different genes have different enhancer regions. You can see that gene A has the dark blue, the light blue, and the green enhancer regions, and the specific transcription factors bind to those. Whereas gene B has a purple one, it also has the light blue element, so this transcription factor regulates both these genes, and it has the red one as well. So different genes involve different transcription factors. So why are these transcription factors so important? Well, we've actually seen them before. We talked about the Hox genes and the Hox mutants previously. Hox genes are transcription factors that control many genes involved in animal development. So here's a Hox gene mutant. We have antennapedia. So here in the wild type Drosophila, it makes little antennae here on its head. If we delete the antennapedia gene, this changes gene expression, and it changes gene expression to develop fully grown Drosophila legs here. So by switching one gene off, we completely change the expression pattern of the region here, and we end up with legs instead of antennae. Again, we've seen this also with the halters here. You delete one gene and you end up with a second set of wings. So this shows you just how many genes these transcription factors are controlling. They're controlling all the genes to make a wing pattern here. So why do we care about this? Well, we, just like fruit flies, have Hox genes as well. And what's cool is if you look at the body patterning and the Hox genes, the similarities between us and flies is very close. So we see another interesting thing here is that the position of the genes in the genome is in the order they get expressed in the organism. So we see here in the little proboscis of the Drosophila, the red gene gets expressed, then the pink gene, then the yellow gene, blue, orange, all the way down to the green genes. Okay, so in humans, we've had some duplications. We have a few extra copies of each of these, but our patterning of the homologs, as they're called, is identical. We have the same positioning there. So studying fly development can actually tell us a lot about our own development because we have this similar patterning. And humans also have Hox mutants. Here's an example of a Hox mutant where this individual, you see what appears to be a normal foot until we get to the middle, and then it's mirrored over here. So this Hox mutation, instead of getting a big toe here, we get a mirror image of the foot over here. So these Hox genes are controlling the patterning of development in animals. Now, I can't leave out the plants, of course. I love the plants. The Madsbox genes in plants are transcription factors that are similar to the Hox genes. And there's 3 different Madsbox gene types, and they control the 4 rings of a flower. So in a flower, you have the sepals, the green protective bits, the petals, the showy bit, the male anthers, and then the carpels, the female bit in the middle. Now, these are controlled by these 3 different genes. A expressed, you get sepals. A and B together, you get petals. B and C, you get anthers. And C alone, you get carpels. What happens if we remove, say, like B? Well, then we get sepals, sepals, carpels, carpels. It affects the development here. And so these master regulators, as they're called, are very important in this role. So transcription factors are super critical here. All right, so let's review. Which of these will bind to enhancers to control the expression of certain genes? Go ahead and pause the video. Think about that. Okay, so it's going to be specific transcription factors binding to enhancers. Those are going to interact with RNA polymerase and increase or decrease the rate of transcription. All right, so in eukaryotes, RNA polymerase needs to bind to the promoter. We have transcription factors, are proteins that influence transcriptional rate. The general transcription factors help RNA bind to that core promoter. And then the specific transcription factors bind to areas outside the promoter on the DNA and enhance or repress the transcriptional rate. The enhancers are the DNA sequences further upstream of the promoter where these transcription factors bind to, and then the DNA bends over, allowing them to interact with RNA polymerase. Okay, we're going to keep working our way down. Now that we have mRNA, we're going to see how that's regulated.